statistics. I know, I feel bad that you have to hold up your hand the whole time. These are the objectives, and I think you got some papers there too, to gain knowledge about normal cell growth, pathways versus mutation of growth-controlled cancer, identify types of cancer staging pathways, and identify cancer prevention strategies and types of cancer screenings. And here we go. So here are some of the statistics that are really interesting. Cancer's second leading cause of death in the U.S., and this was from 2018. Thank you, Karen. The American Cancer Society estimates in 2018 that 609,640 Americans will die of cancer. So these are big numbers. And as you can see, more than 15.5 million Americans with history of cancer were alive January 1st, 2016. Another thing that I want to point out is, like we were talking earlier, people are getting older, people are living longer, and cancer usually comes around a lot of times as you get older. So we're going to be seeing more patients with cancer. Not only that, there's so many ways to help patients maintain their cancer. Some of these cancers are actually like a chronic illness instead of something that's just going to end their life. So there's many more opportunities for patients now that we have. The 5 most common diagnosed cancers are lung, 13%, then breast, colorectal, stomach, and prostate. I gotta flip through some of these things here. Okay, the normal cell growth. So we all have cells in our body and they all have a function. The cell division is tightly regulated. It is a very highly regulated system that works very well. Mitosis occurs for only 1 to 2 reasons: to develop normal tissue or to replace lost or damaged normal tissue. So if something's broken, they'll fix it. Conditions for optimum growth must be present. Presence of specific growth factors, adequate nutrition, sufficient blood supply, existence of inducing tissues, and appropriate space. So all these are very important. They all have a specific function. This is, I'm sure you learned this in school, but we're going to go back to all the fun stuff, the DNA, which we all have, and they are, these are the directions. It's the 4 letters that are used are T, A, C, and G. These represent 4 basics that make up the DNA. The T is for the thymine, A is for the iodine, not iodine, I'm sorry, adenine, I can't pronounce. C is for cytosine, and G is guanine. So they all are paired together, all right? So usually what it is the pairs always are G and T. With the A, these sets are all called base pairs. Those are the base pairs, all right? So there's a certain sequence that we all have. And what happens is sometimes if these sequences come out of order, that can be where an issue lies, all right? So genes are copied many times a day, and with each copy there's a chance of mutation, a mistake. When this goes wrong, a different letter is used. So it's not following the sequence that we have, all right? So what is cancer? A neoplasm characterized by the uncontrolled growth of anaplastic cells that tend to invade surrounding tissue and metastasize to distant body sites. This was supposed to do a little animation, but it's not working. I don't know why. So this got cut off here. These are, I love this slide because it shows you the normal way a cell should look, all right, compared to a cancerous cell. And you can see the difference here. Like here are the normal cells. The large number of irregularly shaped dividing cells is the cancer cells. Here they're all uniformed. Large variable shaped nuclei. Here it's just all kind of in a glob. Variation in cell size and shape. These are usually the normal. They have the same size and shape. These ones would be abnormal. So when the doctors look at the slides, this is what they're looking for on the specific slides. Loss of normal specialized cell features, disorganized arrangement of cells. Here they're all in a pattern, they're just kind of all over. Poorly defined tumor boundary, and as you can see. Pathology of carcinogenic change. Cancer arises from combination of multiple mutations in cell genes. Genomic instability and inflammation. Mutations in regulatory genes. So I like this picture here because it shows you there's a little mutation there. something's going on. Proto-oncogenes, they code for proteins involved in normal cell growth. When mutated, they can enable cancer and be sufficient in growth oncogenes. So, a lot of the mutated proto-oncogenes could be from pancreatic and colorectal cancers. Do you have any questions so far?
No.
Okay. Definitely ask if you want to stop me anytime, seriously. So this is the normal cell growth, okay? So proto-oncogenes encode components of the cell's normal growth control pathway. Some of these components are growth factors, receptors, signaling enzymes, transcription factors. Growth factors bind to the receptors on the cell surface, which activate signaling enzymes inside the cell that in turn activate special proteins called transcription factors inside the cell's nucleus. So we're all back to like biology. The activated transcription factors turns on the genes required for cell growth or proliferation to help to grow and to heal. Oncogenes, gene regulators, are growth that can function abnormally. When proto-oncogenes become overactive, they are called oncogenes and are associated with malignancies. So they're overly active. When tumor suppressor genes no longer function, the tumor suppressor genes are like the stop, it's the brake on a car. If they are not working correctly, cell division is unchecked and cells become malignant. These gene changes are due to damage to the cell's DNA with abnormal activation and deactivation of these important regulatory genes. This is the genetic basis of carcinogenetics. And you can stop me too if you want. This was also a really cool slide because here they're saying the oncogene's off and this one is on, okay, the oncogene. There's something going on here. So this one's gonna say, you know what, this is how we're supposed to do it, this is the normal cell growth, we're doing great. This one, there's an error going on here. So now you don't have that break, that tumor cell saying, hey, hold up, we're making too many. This is like, whoa, we're gonna have a field day and we're just gonna keep producing. All right, so while proto-oncogenes are actually normal genes, an oncogene is an altered gene that had the capacity to contribute to the development of cancer cells. Oncogenes arise from mutated proto-oncogenes. So this is why this can happen. Any little thing, and it's amazing, like we were saying earlier, everything is so perfect in our bodies, the way it's made up. One little thing can throw it off, just like this. This is a great slide that, Karen, that you had, the oncogenes, the different types of oncogenes here, mutations of normal genes, proto-oncogenes that govern growth factors, GF receptors, signal transducers, transcription factors. So here's a whole list, and they tell you the malignancy and the oncogene. All right, so this is something that's in your packet that you can, you know, relate to. I really like this one. So this is the mutation in regulatory cells, and I'm gonna go into more the tumor cells in a little bit here though. The tumor suppressor genes, what they do, they control the proliferation by preventing uncontrolled growth. So that they're the stop, the breaker. There's also a DNA repair gene. They need to correct mistakes. Mistakes can happen, but you know what? It's nice. There's a little eraser that will fix it and fix the mistake. And so they need to correct mistakes caused by carcinogens and replication. Mutated, not functioning, easier to result in cancer. The BRCA1 and BRCA2, breast, ovarian, and prostate cancers. So I wanted to go into, this is another great slide. This shows you the tumor suppressor gene, and then there's the defect. So here you can see this is the way it's supposed to be normally looking, and this is the way that it's not working. All right. So without this stop, without this break, normally our body can say that's enough. I know like multiple myeloma, they'll have like a lot of those proteins that are just expressed. And the estrogen positive breast cancer patients, they're just overly expressed. If they don't have that stop break button, like here it stops it, here it's not. So genetics, we all come from genetics, right? So basically, we all have 46 chromosomes, 22 pairs of autosomes, 1 copy of each parent. So when your parents get together and then there's 2 copies and the sex chromosomes are XX or XY. The genes are the smallest functional units of inherited information. So basically our genes tell us everything, our color, our hair, skin, nails, everything. They control factors for cellular development and function. And they provide instructions for making proteins. A combination of amino acids. It regulates when and where a protein will be produced. So basically, this is like our instructions that we come with. Proteins perform a host of biological functions. Enzymes act to catalyze reactions, support cytoskeleton of cell, play a role in immune responses, and participate in storage or transport of ligands. So this is actually, it says, all cancers are genetic, but as they say here, does not mean that they're hereditary. So we start out as diseases of the genes, okay? This does not mean it's all hereditary. Since not all genes are expressed in every cell, an inherited defect does not ensure that a cancer will develop in any one person. When you think about it, there are some families, someone may not have smoked their whole life and they end up getting with lung cancer. Or you have a family member that smoked their whole life and didn't get lung cancer. A lot of these are related to the genes and how they're expressed and the defaults that happen within each one. Most occur sporadically due to environmental factors that cause DNA damage. And I'm going to go into environmental factors in a little bit. Growth factor receptors, mutations, and GFRs cause multiple copies of gene to be made. They're amplification. They're making multiple, like those pictures that we showed earlier. This leads to an overexpression of cellular proteins, HER2, basically with breast cancer patients, and actually prostate, I want to say. There's an overproduction of the HER2. There's no stop button. They're just going on and on. Greater degree of amplification is corrected with poor prognosis. Common hereditary cancer syndromes. In the case for genetic testing can be covered by insurance, but there has to be a reason for genetic testing. They're not going to just do genetic testing on anyone. There are clear indicators of when they can do genetic testing, unless you want to pay out of pocket. So the indication, there's criteria, varies depending on the gene tested. The universal is they have to have a confirmed family history. Informed consent, you always have to have consent, tests that can be interpreted, and not everyone's appropriate. I know working at Pluta, our breast cancer patients, if they're over the age of, I want to say 70, they don't really do, because at that age they're like, well, we're not really going to do genetic testing. However, if their mom and their sister were diagnosed like at a higher stage, that would be one indicator, but a lot of times over the age they won't really do the genetic testing.
Don't you have to have cancer to be
No.
eligible? No. Okay.
No, and actually I think like if they could check for the BRCA2 and stuff like that, for patients that, their mother and grandmother had breast cancer.
Mm-hmm.
And then that patient can do genetic testing.
Mm-hmm.
But as I said though, some people can do genetic testing, but it might not be covered in insurance unless they follow the certain criteria.
They're getting much more lax. We're testing much more than we used to be able to. And with breast cancer, if you're premenopausal, there's a good chance, even if you don't have much of a family history, there's a good chance they will want to test you. The ideal is that someone, you ask if you have to have cancer to be tested. The ideal is that you have tested someone with cancer so you know what mutation, if there is a mutation. So if, I'm over 40 now, but if I were under 40 and was concerned because maybe I had a lot of family history, especially younger age, but none of them had been tested, they would still test me, but that's where you can end up with tests that aren't clear, because if they don't know exactly what they're testing for. But if say my mother had been young and she had been tested and they found a mutation, now they know exactly what mutation to look for in me.
Okay.